Hello, my name is Gabriela Imre, and I am working at the live cell imaging facility at Karolinska Institute in Sweden. In this short video, I will share with you some tips regarding sample preparation. The most important points when preparing samples for microscopy are the following. Measure the fluorophore with the microscope filters. Use cover slips of thickness specified as number 1.5, meaning 160 to 190 micrometers. Put the sample on the cover slip, not on the slide. Do not cover the slide ends with the cover slip. Allow mounting medium to harden or use nail polish to completely seal. Seat cells evenly and not overcrowded. Use the maximum volume of medium for live cell imaging. Use fresh paraformaldehyde. Now we will go through all these points one by one. When deciding which fluorophore to use to label your sample, you must first make sure that their spectra matches the filters available on the microscope you will use to image. Watch the bleed-through video found on our website to understand how to do it. Google LCI facility to get to our website, and under Learning Microscopy, click on Teaching Material. You will find the video there together with other useful videos. The cover slip on your sample is part of the design of the objective. All objectives from all manufacturers are designed for 170 micrometer thick cover slips and with the sample placed directly against the cover slip. All cover slip manufacturers follow the same label system. Cover slip label as number 1.5 have a thickness of 160 to 190 micrometer. Cover slips labeled as number 1.5H have an even narrower range of thickness, 170 micrometer plus minus 5 micrometer. Number 1.5H cover slips are more expensive. Use them when doing turf, super resolution, and when using very high numerical aperture objectives. Never use cover slip labeled as number 0 or number 1 since they have the wrong thickness. If you use cover slip with the wrong thickness, you will not be able to get the best focus possible when imaging. When purchasing multi-well plates, check the height of the skirt at the bottom of your plate. The skirt is the plastic edge that leaves the bottom of the plate so that the glass doesn't touch anything when the plate is on the table. When you want to image the wells at the edge of a plate with high skirt, the objective might bump into the stage insert. It is dangerous for the objective and the focus will be lost. In many cases, you will then need to exclude all the outer wells of your plate, leaving the plate with 60 wells instead of 96. So please do not buy multi-well plates with high skirt. Most people who cut frozen or paraffin-embedded tissues place them on a thick glass slide after cutting. Then they add mounting medium and the cover slip and image through the cover slip and mounting medium. This is not the correct way to prepare a sample. All objectives from all manufacturers are designed for a sample where the cover slip is 170 micrometer thick and the sample is directly touching the cover slip. Then you place your sample on the slide. The mounting medium ends up between the objective and your sample. This means then you introduce light scattering. Scattering will prevent you from getting the best focus, and the amount of light that reaches the objective will be reduced, so images will be both dimmer and blurrier. Instead, you should place your sample on the cover slip as shown here, so that when you image, the mounting medium is not between the objective and the sample, and you will have the best imaging conditions. It is easy to put sections on cover slips instead of slides. We will show you how to do that now. You should also watch the video about cleaning cover slips to increase the adherence of your sample. The procedure of applying your paraffin section on a cover slip follows the same steps as putting your section on the slide. As you can see, the paraffin block is sectioned and processed as usually. But now, instead of a slide, she will use a cover slip to put the section on. You can also add several sections on the same cover slip, as you will see in a few seconds. The cover slip fits in the racks usually used for the slides. Cytospins were designed to spin the cells on the slide, but as we explained earlier, this is not the correct way for imaging. Instead, you should cytospin them directly on the cover slip. Use 2 small pieces of tape to attach a clean cover slip to a slide, and use the slide only to hold the cover slip in the cytospin. This way, the cells will land on the cover slip. You will only notice that the focus is not acceptable until you compare the two samples side by side, the one with the cells on the slide and the other one with the cells on the cover slips, as shown here. The cells on the top are on the slide, and under these conditions, this was the best focus we could achieve and the best image to acquire. The cells on the bottom are on the cover slip, and you can clearly notice the difference in the quality of the image, sharper and brighter. Do not cover the ends of the slide with the cover slip. When using an inverted microscope, the sample on the left, when put on the microscope stage insert, will rest on the slide on one side and on the cover slip on the other side, so it will not be flat. Each image will show a focus gradient. The sample on the right will rest on the slide only on both sides. The focus will be more even across each image. The mounting medium will protect your sample from bleaching, but make sure it does not end up on the objective, as it will damage it. Remove all PBS from the last wash before adding mounting medium to avoid diluting it. If you use hardening mounting medium, allow it to fully harden on a flat surface before imaging. If you use non-hardening mounting medium, completely seal the cover slip with nail polish and allow the nail polish to dry properly. Remove PBS salts by cleaning the outside of the cover slip with a drop of water. When you prepare your cells for immunostaining, the first step is fixation. Only use fresh paraformaldehyde to fix your sample. Buy the smallest quantity of paraformaldehyde powder available. Make a working stock solution. Allocate most of it in 50 ml Falcom tubes and some in 1 ml tubes and freeze down. Keep a small quantity in the fridge as concentrated working stock, but for max 1 month. If you buy formaldehyde solution instead of powder, buy the highest concentration possible. Do not keep it in the fridge, as it will degrade with time. As soon as you receive it, aliquot it and freeze it as mentioned above. Only keep a small working stock in the fridge. Be aware that most commercial formaldehyde solutions contain 10 to 15% methanol. Your antibody might not be compatible with it. As you can see in this image, the paraformaldehyde used to fix these cells was not good. One sees that the cells are partially detached and folded on themselves. Here we summarize again the most important points to know when prepare samples for microscopy. Thank you for your attention.